Welcome to the topic lecture on sexual reproduction, meiosis, and fertilization. Here are the objectives for this topic. As always, I expect these to be mastered by the time we have our next test. Please let me know if you need any help with these objectives. In this lecture, we are going to discuss sexual reproduction, meiosis, and fertilization. Organisms reproduce primarily in two ways, asexually or sexually, and sometimes both. Asexual reproduction is similar to the process of mitosis we discussed in the last chapter. An organism that undergoes asexual reproduction will create an offspring that is identical to the parent. This is beneficial for a host of organisms because they have already arrived at a design that is successful in the environment, and by creating a new, exact copy, the offspring is likely to be successful in the environment. Sexual reproduction is significantly different, starting with the fact that two individuals, in general, are needed to create an offspring. Through the process of sexual reproduction, the genes of these two parents are shuffled and create a new, unique individual. This individual may be more, equal, or less successful than their parents in the environment. This variation within the population is one of the keys to evolution and allows a population to adapt to any changes in the environment. Another aspect of sexual reproduction is filtering by the population pool. By making reproduction a competitive event, only the most fit individuals are likely to pass on their genetics. This ensures that those who have mutations that are weaker are unlikely to continue passing on these traits. This should be review for you, but let's review it again. A diploid cell is a cell in an organism that contains the full complement of DNA for an organism in a 2 times or 2x system. Some of these diploid cells serve as somatic cells within the body and don't contribute to the production of offspring. But some diploid cells are dedicated germline cells. These diploid cells are charged with the production of the gametes, and while they don't specifically form the new offspring, they do create the cells that create the offspring. The cells directly involved in offspring production are gametes or germ cells. These cells have half the DNA of the diploid cells and are known as haploid cells. This makes sense because if two cells have come together to create the new individual, you would want the original cells to have only half the DNA. Otherwise, the zygote would contain double the DNA, and that would lead to fatal issues, as we have seen in previous chapters. On the surface, meiosis appears to be the running of mitosis twice, but with skipping interphase and DNA replication, and for the most part this is correct. The chromosomes are lined up and divided twice in similar process to those that we learned about in mitosis. However, there are some key differences between meiosis and mitosis. One difference is that the products are different. In mitosis, the product is 2 identical daughter cells, while meiosis generates 4 unique daughter cells with half the DNA. Meiosis can last a very long time. For example, in human females, their eggs undergo meiosis 1 during development, but wait to undergo meiosis 2 until they reach puberty. This causes the entire process to last decades, whereas mitosis does not last that long. The biggest difference between meiosis and mitosis is the handling and splitting of the chromosomes. How chromosomes are separated is one of the defining differences in the generation of gametes. Just like in mitosis, the chromosomes are duplicated once in S phase, creating sister chromatids. In mitosis, these sister chromatids are separated after metaphase, with one of each sister ending up in the daughter cells. However, in meiosis 1, the sister chromatids stay attached, and instead the replicated chromosomes are split. This means that after meiosis 1, the 2 daughter cells still have 46 chromosomes, but these are duplicated sister chromatids and thus are not considered diploid cells anymore. They are haploid cells. In meiosis 2, these sister chromatids will split, leaving each cell only 26 chromosomes. This division is further This division is important because it helps create some of the genetic variation we have talked about by creating a cell with only half the DNA needed for a new individual. On the next slide, we will discuss how this variation is further achieved. We have talked about recombination before in Unit 2 when we looked at DNA repair. In this instance, we are evaluating the use of this as a means of generating genetic diversity. This process is known as crossing over. As you know, chromosomes contain genes for various traits, and each pair of chromosomes have the same gene in the same position. However, the exact coding in these genes may vary slightly, so when crossing over occurs, the chromosomes will still maintain all the same genes, but the information within the genes may now be varied. It's like putting on outerwear. You have a coat, gloves, and hat, but then you switch out your hat for a slightly different hat. You still have a hat, but your outfit is now different. This whole process is facilitated by a protein machine known as a synaptonomal complex. This machine helps hold the tetrad of chromosomes together to help create the crossing over sites known as chiasma or chiastoma, which is the plural. The tetrad of chromosomes frequently has between 2 to 3 chiastoma and hold the tetrad together and help drive chromosomal sorting we have previously discussed. So how do these chromatids end up separated and in their final cells? It's similar to the process of mitosis in the fact that these chromatids are separated using kinetochores, but there are significant differences in the process. Remember, the sister chromatids stay connected throughout meiosis 1. This is dramatically different than what we see in mitosis, where the sister chromatids separate first. These sister chromatids are held together by the cohesions we discussed in Chapter 18. When anaphase 1 is reached, the cohesions at the end of the chromosomes are degraded, but those near the center of the chromosomes are held together. These kinetochores on the sister chromatids function as a single unit during meiosis 1. In meiosis 2, the sister chromatids then separate. This happens because the cohesions in the center of the chromatids are now degraded. Kinetochores function on their own, allowing each chromatid to be pulled in its own direction. This leaves the cell with only one sister chromatid for each chromosome. Take a moment to review the differences between meiosis and mitosis. What are the key differences? In summary, in meiosis, 2 cell divisions are required after DNA replication to produce the haploid gametes. Each diploid cell that enters meiosis therefore produces 4 haploid cells, whereas each diploid cell that divides by mitosis produces 2 diploid cells. Although mitosis and meiosis 2 are usually accomplished within hours, meiosis 1 can last days, months, or even years because of the long time spent in prophase. Unless you are an identical twin, no individual will ever have the same genetic code as you. Even your siblings' genetic codes are variable and may differ slightly from yours or may be dramatically different. How does this happen? It's still your mom and dad's gametes. Shouldn't these gametes produce the same offspring every time? Nope. Remember, we just talked about how crossing over will create unique chromosomes that are in each gamete. That means that even if you and I only had one chromosome from our parents, we would still see dramatic variation. Remember, this crossing over occurred in your parents, so the genes that were mixed were actually those of your grandparents. Beyond this crossing over, each parent has 2 unique chromosomes in each pair. These chromosomes sort independently of the other chromosomes, so each egg or sperm will end up with its own combination of your grandparents' chromosomes. That is why no matter how many siblings you have, none will ever have the identical genome you have. It is important to note that sometimes chromosomes don't separate like they should. This event is known as non-disjunction and can result in gametes with too many chromosomes. Some of these errors, such as Down syndrome, can still support life, while other chromosomal duplications are not compatible with life. These errors usually occur in the creation of eggs rather than sperm, as sperm have a higher selection criteria during development. Those gametes that do become fertilized and result in too many chromosomes in the zygote oftentimes end as a miscarriage during early pregnancy. This whole process starts the production of a new individual when a sperm and egg fuse together to combine their DNA. When the first sperm penetrates the egg, a calcium, I know you missed calcium, release is triggered that hardens the outer shell of the egg, known as the zona pelliculia, and prevents further sperm from entering the egg and contributing more DNA than a zygote would need. Fertilization is considered complete when the nuclei of the egg and sperm fuse. This is the end of this topic. Let me know if you have questions about this information or if you would like to go over the topic again.